Hey Chutkus, I welcome you all to second PU Midterm revision series, and this will be for biology. So we saw a lot of children demanding for please do midterm series, so we thought we should be fulfilling it, and I thought I'll take 3 chapters at the same time, okay, so that it will be helpful for you to study everything in one go. So human reproduction, reproductive health and sexual reproduction in flowering plants are the topics. So reproduction is common among all of them, right? And here in this particular video, we'll be talking about the most expected questions that could be coming. So we have analyzed the previous year question papers, and also we have taken certain questions from the model question paper that was put down by government. So you'll get to know what type of questions could be framed, or what type of questions were framed, and what can you expect, and how can you write the answers for the same as well. Okay, so going ahead, let's see the question paper pattern. Children, you will be having 6 parts. Okay, part A is divided into 2, rest all of them are significant parts. So part A will be having A1 will be having MCQ questions. 15 number of questions will be there. If you attempt all of them, you get 15 marks. Okay, you can attempt all. Now part A2 will be having fill in the blanks question. 5 of them will be there. So fill in the blanks will be like there will be sentence with a blank and you have to fill the blank area, but they will be giving you options as to what has to be filled. Okay, so that is there. I'll show you a question like that, and you have to attempt all. So if you attempt all, 1 marks for each, you get 5 marks. Now part B will be having short answer questions, so 2 marker questions. 7 questions will be there. You have to attempt at least 5 of them. Only 5. Okay, so 5 into 2, 10 marks. Fine. Now over here part C will be having again short answer type questions, but this will be of 3 marks. Again 7 questions will be there. Out of that you have to attempt 5. So 5 into 3, basically 15 marks. Okay. Part D will be having long answer 5 marker question. So over here 7 questions will be there. You have to attempt at least 4 of them. 4 out of 7. So 4 into 5 is children 20 marks. Yeah. And part D, the 6th part that will be there, it will be again having the long answer type question, 5 marks. 3 questions will be there. Out of that, at least 1 you have to attempt. Okay, so 1 into 5 is basically 5. The total becomes 70. So your question paper will be out of 70 marks. Okay, so I said just this A is segregated into 2. No, this is also segregated into 2. But yes, this matters and you have to know this. Please do not go on attempting all the 7 questions in these things. Otherwise, your timing will be gone like this. Yeah, so that's why I made sure I was telling. Always remember, in 3 marker, 2 marker, 5 marker, everywhere you are given a choice to attempt these many number of questions. Yeah, now let's start. So what is the weightage? Human reproduction will be giving you 10 marks. Okay, reproductive health will be giving you 6 marks, and sexual reproduction in flowering plants will be giving you 11 marks. So in total, if you see, this has the highest number of weightage. Then comes human reproduction, and then comes reproductive health. Cool. Now what type of questions can you expect from each chapters? Children, from human reproduction, there will be 3 marker. For sure, there will be a 3 marker question and there will be a 5 marker question. I didn't mention about MCQs and everything because you know, anyways, you have to study, you have to tackle with MCQs. So MCQ questions will be there for all the chapters, that is there. But I'm specifically saying a 3 marker question will be definitely there from human reproduction, and a 5 marker question will be there from human reproduction again. So reproductive health would not be giving you 5 marker question, but it will be giving you 3 marker question. That is the highest weightage. Okay, that will be giving you 3 marker, and apart from that, it can give you MCQ, such that in a way 6 marks will be given to this chapter somehow. Okay, so 3 over here, rest of the things might be in 2 or 1 or things like that. Now sexual reproduction in flowering plants. Over here again, you will be having a 3 marker and a 5 marker. Why I am not mentioning about MCQ even though it was given in their blueprint? Because I want you to study really hard. Okay, please study. Do not say that, okay, what's the use of me telling that, okay, definitely MCQ will come. Anyway, you have to study, right? That does not bring any change. But when I say that 5 marker or basically 5 marker or 3 marker questions will come, probably you might try to write things and understand how you have to frame your answer, big answers. So which is why I have mentioned over here. So you can expect 3 and 5 from both these chapters, and 3 marker from reproductive health, and might be 2 marker will be there. MCQ might also be there. Cool. Now let's see the first type of question that we have over here, that is from human reproduction. So choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. So you have to find what is responsible for the formation of what? This particular label part. And what is this label part, my dear children? It is zona pellucida. It is what? Zona pellucida. So your options are, it is formed from the primary oocyte before fertilization, primary oocyte after fertilization, secondary oocyte before fertilization, secondary oocyte after fertilization. So you have to know the answer. How can you understand this term? Very easy, very easy. So we have to understand how is ovum being formed, or what is secondary oocyte, or what is primary oocyte for the first time. Okay. So basically inside the ovary you have so many oogonia. What do you have, children? You have so many oogonia. How many? Millions of them. And from them, some of them degenerate, they just die off, and rest of the cells, they say that we want to become ovum. So basically they start undergoing a type of cell division which is known as meiosis. Okay, and what exactly happens in meiosis? Suppose this is a cell, this cell will divide into two. Okay, again what will happen? These cells, the daughter cells, they will also divide into two. And now we say that the meiosis has got completed. So what exactly happens in meiosis is that you take a cell which has 2 n number of chromosomes, that is double the amount of chromosome. Okay, you divided it and now it has half the number of chromosome. It will be having half the number of chromosome when you divide it for the first time. The second time it will be having the same number of chromosome just like the parent over here. So in a way, when we started off, we had 2n, but the end, what are we getting? Just n, that is half the number of chromosome. This whole process is known as meiosis, or you call them reductional division. So always understand, when we want to form gametes, okay, ovum is basically a gamete, right? So when we want to form a gamete, it has to undergo meiosis, because why, children? The gamete should be having half the number of chromosome. So from the ovum, there will be half number of chromosome. From the sperm, there will be half number of chromosome, and together you will be finding half and half come together and form 2n. So n and n comes together to form 2n number of chromosome. So if both the parents are giving every chromosome to us, then probably we will be having more number of chromosome, and we do not want that. We just want 23 pairs of chromosome or 46 number of chromosome, that's all. So you have to halve the number of chromosome, and this process of cell division is known as meiosis, where meiosis has two phases. This is phase 1 of meiosis and this is phase 2 of meiosis, and when they complete the whole process, we say meiosis is done. Okay. Now in meiosis 1, there is prophase, there is metaphase, there is anaphase, and there is telophase. And since this is occurring in the first phase, you say P1, M1, A1 and T1. Now similarly, it will undergo meiosis 2. That is phase number 2. So over here also it will be prophase 2, metaphase 2, anaphase 2 and telophase 2. Why is ma'am telling all about this? Because I want to teach you about the oogenesis over here. With this, that concept is clear, right? Now over here I am saying that this oogonia has started undergoing what? Meiosis, and now I'll change the name of oogonia into something else. I'll call it primary oocyte now. I'll call it primary oocyte. But there is a problem. Primary oocyte, when it started getting divided, it was in a phase that, okay, at last I'll complete my division and form the ovum. But it got stuck over here. It could not go ahead on the metaphase 1. So primary oocyte basically gets stuck in prophase 1. Prophase 1. Okay, and when it is stuck, there are certain cells that comes to console it. So you have primary oocyte over here. Okay. And this primary oocyte basically gets surrounded by certain cells which comes to console it. You call these cells as granulosa cells. What do you call them? You call them as granulosa cells, and together the primary oocyte and the granulosa cells together, what do you call them? So I am writing primary oocyte plus granulosa cells, you call them, this is the primary follicle. And the primary follicle, this is about when we are in the embryo, when we are forming in the mother's womb. So at this point of time, all this has happened. But now what happens is that once we are given birth, still we are having primary oocyte and it is surrounded by granulosa cell, still it is primary follicle, because the primary oocyte is getting arrested in which stage, children? P1 stage, prophase 1 stage. Now what will happen? Slowly, okay, when we hit puberty. So until we hit puberty, we are in this stage only. All are in our ovary, we will be having so many primary follicles having the primary oocyte. Once we hit puberty, during this point of time, almost we are left with 60,000 to 80,000 primary follicles. Rest all of them might have degenerated again. So initial degeneration took place here in the ovary itself, embryonic ovary. Now over here also, okay, when we grew up, until the time we reached puberty, only this many are left in each ovary. So this is left in one ovary. These many number of primary follicles will be left in another ovary as well. Okay. Now what has happened? Once we hit puberty, there is another thing that happens, that is the primary oocyte which is there, it's still like that only, but along with the granulosa cells, there is one more layer coming to console our poor primary oocyte, and that layer will be known as theca layer, and that layer is known as, children, theca layer. Fine. And this whole structure where granulosa cells plus theca plus primary oocyte is there, I will now call this structure as secondary follicle. I will call now this as secondary follicle. Okay. Now what will happen? This secondary follicle will later say, I want to become tertiary follicle. So what is there in tertiary follicle? The same thing. Again over here, I am just going to erase this. Okay, and I am going to make the primary follicle. So over here I have the primary oocyte surrounded by the granulosa cells, and now the theca layers will be different. So there will be one theca over here. I will say this is theca interna. Okay, I will say this is theca interna, and there will be one layer outside this theca. That is also a theca layer, and since it is on the external surface, I call it theca externa. Okay, one more change has happened, that is, in between the granulosa cells there will be an area which will be fluid filled space. This is an area which has fluid filled space. This area is known as antrum. So initially we had primary follicle. When the primary follicle basically had theca layer around it, you called it secondary follicle. Inside it you have the primary oocyte only, okay, which is stuck in prophase 1. Over here it has not completed division, okay. But now it has to form tertiary follicle. When puberty hit, you formed secondary follicle. After that, now you are going to make tertiary follicle. The changes that comes in tertiary follicle is that the theca also gets divided into inner theca interna and outer theca externa, and there will be one cavity, that is antral cavity. Now the oocyte which is there inside says, I want to divide, I want to complete at least my first meiotic division. So it completes its division, and now you will get 2 cell. Okay, now you will get 2 cell. The one cell you call it as, okay, so I am going to bring out certain changes over here. So this cell has divided. Now we have got 2 cell. One cell will be big, other cell will be small. Okay, one cell will be big, other cell will be small. The big cell I call it as secondary oocyte. What do I call it? I call this as secondary oocyte, and the small cell I call it as first polar body. I call it as the first polar body. So inside the tertiary follicle, what do I have, children? I have the secondary oocyte, okay, and I have the first polar body. The first polar body says there is no use of me, so I'll just go. So this gets degenerated, okay. Now we have to deal with secondary oocyte and the tertiary follicle. So always remember, in the secondary follicle you have the primary oocyte, in the tertiary follicle you have the secondary oocyte. Now, when we say that each month one particular follicle is being released and it matures to basically release an ovum or egg, whatever it is. So basically what is happening is that the secondary follicle is basically maturing into tertiary follicle, and the tertiary follicle, because of the hormones, are maturing into the next type of follicle, which is the last type of follicle that you are having, which is known as Graafian follicle. What do you call them, children? Graafian follicle. So I'm not going to make the entire structure of Graafian follicle, but I'll tell you gist of it, okay. So what happens in Graafian follicle is that, so from here, so you know theca layers are there, so I'm not writing it. Theca layers are there, I'm writing it over here. Theca layer is there, your antrum is there, okay. What happens in the Graafian follicles, that is changing from the tertiary follicles? The antral cavity becomes bigger. The antral cavity becomes bigger, and there is one more change, okay, there is one more change. You remember the secondary oocyte which is there, and the polar body, it went off, but the secondary oocyte will be now forming a layer around it. Secondary oocyte itself will be forming a layer around it, my dear children, and this particular layer is known as zona pellucida. So who is making this? The secondary oocyte is making this. So over here we have come until this particular stage where one cell is being formed, that is the secondary oocyte, and the other cell is the polar body. So obviously the polar body is lost. So if I say that this is the first polar body, then I cannot expect these things to happen over here. So this set of thing is gone. Now the secondary oocyte is there. Secondary oocyte, when it completes a division, that's when you get ovum, and then this cell, what happens to this cell? This is the second polar body that will be released. This also will be degenerated. So at the end you will get this. But as of now, it has now divided into secondary oocyte, and secondary oocyte is trying to divide itself, but it has got stuck in metaphase 2. So initial stuck was at prophase 1, that was by primary oocyte. This secondary oocyte is getting stuck in metaphase 2. So over here I am saying this particular secondary oocyte is getting stuck in metaphase 2. Here also it is still stuck in metaphase 2, and to console it, it makes a layer around itself that is known as zona pellucida. Okay, that is zona pellucida. Now zona pellucida, outside that also there will be certain cells. Over here granulosa cells again comes over here and says that we will help you out. We will help you out. But these granulosa cells have a separate name. You call them corona radiata. You call them corona radiata. This is not made by the secondary oocyte, okay? This is not made by secondary oocyte. These are the set of granulosa cells. The one that is made by secondary oocyte is zona pellucida. The only layer that it makes is zona pellucida. Now outside it, as I said, antral cavity will become bigger. So over here you will be having the antral cavity, and then you will be having your whole granulosa cells again. Okay, you will be having your granulosa cells, and you will be having the theca layers around it. So this thing you have to remember. Later on when the sperm comes and attaches, okay, when the sperm comes and attaches over here, that's when the secondary oocyte will finally complete its division to form the ovum. Okay, so primary oocyte before fertilization, does it make it? No. Primary oocyte has nothing to do. Primary oocyte after fertilization, does it make it? No. Secondary oocyte before fertilization, does it make it? Yes. Secondary oocyte after fertilization, no. It is done before fertilization. Because why is it actually doing it like that? Because secondary oocyte says, now which I waited for this long. Now whatever sperm has to come basically has to get into this particular chakra that I am making around myself, and not all sperms can come and attach there or get inside the ovum because of so many reactions that we will be talking about today itself. Okay, so basically this is a chakra layer that is formed by the secondary oocyte itself before fertilization. So this is the answer for this. Why did I explain all this to you? Because you can expect so many questions from oogenesis, or you might be asked to explain oogenesis itself. So you should be knowing about that. That's why. Okay, works. Now going ahead, children. Out of the options given below, choose the correct stage for the transfer to fallopian tube for successful IVF. What do you mean by IVF? In vitro fertilization. Okay, so what happens in vitro fertilization, children? What happens in vitro fertilization? Well, this is not of human reproduction, this is of reproductive health. Okay, but yes, you should be knowing, so all these things are linked. So basically, the fertilization, okay, the fusion of sperm and the egg is not taking place inside the human body in the case of in vitro fertilization. Okay, in vitro fertilization means in the laboratory condition you are going to do fertilization, that is outside the body. Okay, so in this case the sperm is taken from the donor, the egg is also retrieved from the female donor. Okay, the sperm and the egg basically are made to fuse. Okay, they are made to fuse in the lab, and now they are given all the growth medium and everything such that you got the zygote, no? The zygote will obviously divide itself to form the embryo. What is a zygote, children? When the egg and the sperm fuse, one cell is formed. Okay, together one cell is formed. The egg is basically there with the nucleus and everything. The sperm comes just with the nucleus. The sperm just has the nucleus and the mitochondria, but mitochondria everything goes off. Just the nucleus is given to the sperm, so is given to the egg. So basically egg is the one that contributes to the cytoplasm and everything apart from the nucleus from the parent, from this parent. Okay, so this nucleus comes, fuses, you get just one cell, and that one cell is known as zygote. This one cell, when it start undergoing division, now when it is 2-celled, it's 4-celled, or it is 8-celled, all the time you call this as embryo. But embryo, since it divides, I cannot insert the embryo at whatever stage I want. Okay, Obviously, I cannot make the fetus in the lab itself. I want. In the case of IVF, what do? You make the embryo in the lab, or you make the zygote in the lab. Then later, you put it inside the fallopian tube of the female. So, you remember the fallopian tube? Okay. So, over here, in this case, you have two options. Okay. Either you can put this thing in the fallopian tube or in the uterus. But there is a challenge. If you want to put the embryo into the fallopian tube, then the embryo should be having just up to 8 cells. Just up to 8 cells. So, embryo should be having 8 blastomeres. 8 blastomeres means 8 cells. If it is more than 8 cells, then it should be put inside the uterus, children. Okay, it should be put inside the uterus. Fine. So, basically, over here, when I say only up to 8 blastomeres, you can put it up here. Then later on, if you want to put the embryo, if the embryo has divided itself more than 8 blastomeres, then you should be putting into the uterus. Then only implantation will be taking place. Fine. Fine. Now, if you think that you are putting 16 blastomere embryo into over this area, you will lead to a condition which is known as ectopic pregnancy. It will lead to a condition which is known as ectopic pregnancy. What do you mean by that? Pregnancy at the place other than the uterus. So, this point of time, the mother and the fetus both will be dying. Okay, because it is not the space for the baby to grow. So, over here, when I put the embryo up to 8 blastomeres, this embryo will slowly divide itself and then roll them down into the uterus later on by itself. It's a normal process that will be happening. Okay, so this is the correct answer, children. What do you call this process? As zygote intrafallopian transfer. Okay, when you're putting the zygote inside, you put zygote intrafallopian transfer. When you're putting the embryo inside, you call it embryo transfer. Okay, so when you're directly putting the zygote, you call it zygote intrafallopian transfer. When you're putting the embryo, you call it embryo transfer. That's all. Works? Going ahead. Apomixis is the production. You have to find what? Basically, it is an assertion reason question from sexual reproduction in flowering plants. Okay. Now, statement 1 says apomixis is a production of seeds from unfertilized ovule. Is it true? Is it true? What do you mean by apomixis? So, usually what happens, I'll tell you. Usually, you have the ovary. The ovary has the ovule. Okay. And the ovule have 3 set of cells over here. 3 set of cells over here and 2 nuclei over here. One cell that is over here, it is the egg cell. It is the female gamete that we have. It is the egg cell. So, when the pollen grain comes, it carries the male gamete, and now this and this fuses, and now this particular thing that you see over here will transform the whole ovule into a seed. When the fertilization of the male gamete and the female gamete takes place, at this point of time, what will happen? The ovule as a whole will turn out into a seed. And this particular thing, the egg cell plus the male gamete will form what? The zygote inside. The zygote will later divide into embryo. But it is all happening inside the ovule. That is, now you call this ovule as seed. Okay. Now, over here, apomixis is a production of seeds from unfertilized ovule, as in fertilization has not taken place. Then how can you produce seed? How can you produce seed if fertilization? I told you only if fertilization has taken place, that's when you say ovule is now a seed. Now I am saying apomixis is a production of seed from unfertilized ovule. Is it okay? How can you produce seed without fertilizing? Is it okay? You might think it is not possible, but this is actually true. And what is it? This process is known as apomixis. This process is known as apomixis, children, where you are producing a seed, okay, where you are producing a seed without fertilization. Without fertilization. So, how does it take place, ma'am? That is the first thing that you should be knowing, okay. So, over here, as I said, this is the ovary. Assume that this is what? The ovary. And over here, assume that this is the ovule. Cool. Now over here you have, I said 3 cells. One cell among them is what, children? Is your this thing, that is the egg cell. Rest all of them, both of them are synergids. Okay, what do you call them? I will explain you here itself. You call them synergids. Fine. Over here you have 3 cells again. These 3 cells you call them antipodal cells. You call them antipodal cells, and then you have 2 free nuclei over here. 2 free nuclei. So what happens is that all of these cells, all the cells that you see over here, usually they are a result of meiosis. Okay, usually not exactly the result of meiosis. Basically what happens is that you have a cell, okay, initially this particular ovule, it looked like this, where you had so many cells, and all the cells you called it nucellus. Okay, you call them nucellus. Now one among them, okay, one cell among them said that I want to undergo division and help in the formation of, let's say, the female gamete. So this particular cell over here, let me call it megaspore mother cell. Megaspore mother cell said that I want to divide myself. So this cell underwent meiosis. So when I say meiosis, so initially it had 2n number of cells, it underwent division. Now you have n, half the number of cells. Again, meiosis is in 2 parts. So you have other 2 cells. Here also you have other 2 cells. All of them have n number of chromosomes, right? So from one megaspore mother cell, you should be getting about 4 cells. But what happens is that these 3 cells gets degenerated. One cell stays there, and this one cell that is being formed, this is a megaspore that is being formed, it later transforms into something that is known as embryo sac. This one megaspore is the one that will form the embryo sac that looks like this. Okay, there will be an embryo sac that looks like this. What exactly happens in the embryo sac? So for suppose this is an embryo sac where I had this one cell's nucleus, okay? This one cell, the whole thing that you see over here, this is an embryo sac, right? This embryo sac with just one nucleus, it will now divide itself with the help of mitosis. Mitosis is different, meiosis is different. Meiosis is this. Mitosis is basically when you have a cell, whatever number, for example, here n number of cell was there, right? N number of chromosome was there. So when I divide, I'll still get n. This is known as mitosis, where I just want to divide into 2. So over here the nucleus divides into 2. Okay, again this nuclei divides into 2. Again this nuclei divides into 2. So basically you have 3 rounds of mitotic division. You have 3 rounds of mitotic division. So first when you divided, you got 2 cells. Next you divided, you got 2 cells from here, 2 cells from here, that is 4 cells. Now when you divide again, you will get 8 cells. So at the end, how many cells will be there, or how many nuclei will be there? The nucleus is only dividing, the cells are not dividing as of now. So you have 8 nuclei inside the embryo sac. You have 8 nuclei formed over here with the help of what? Embryo sac. So do not get confused. One megaspore will be forming an embryo sac. An embryo sac is nothing, it just has one nucleus. This one nucleus will undergo mitosis. Now all the nucleus that we see will be having half the number of chromosome, that is n number of chromosome. And now the nucleus is surrounded by cells, and now it will look like this. So 3 cells over here, 3 cells over here, 2 nuclei are not surrounded by anything, they're just randomly floating around there. So how many cells do we have? 1, 2, 3, 4, 5, 6. 6 cells and 2 nuclei. At the end, how many nuclei did we have? 8. Similarly, so 6 plus 2 is also 8 itself. So usually it happens like this. So all of them should be having n number of chromosome. Agree? It should be having n number of chromosome including the egg cell. And now when the pollen grain comes with the male gamete will also have n, and n comes together forms 2n. Everything is all cool. But this is not normal in the case of apomixis. In the case of apomixis, it is said that the seeds basically somehow didn't undergo the process of halving the number of chromosomes. Rather than that, all of them, they already have 2n number of chromosome. They already have 2n number of chromosomes. So if already 2n is there, do I need pollen and anything? No. So do I need fertilization? Nothing. This egg cell slowly will transform itself. Okay, this egg cell will slowly transform itself into what? A zygote. without fertilization. So it is already having 2n. All of them that you see over here are already having 2n. This egg cell also. So basically this egg cell without undergoing fertilization is now going to form zygote, and when it is forming zygote, the process of fertilization also involves the process of forming zygote. That's when you call it as a seed, right? So since it is forming a zygote and then an embryo, I can still call it a seed. I know this particular process is so much like it flips your brain around, but this is how it is, okay, and you have to learn like this only. So that is one step, okay. This is known as apomixis. This is one type of apomixis, children, okay. This is one type of apomixis. Now you tell me the product that will be got, okay, the seed that will be got, or whatever plant will be arising from this particular seed, don't you think that will be an exact replica of the parent? It will be an exact replica of the parent, right? Right. So over here, why it is an exact replica? Because fusion has not taken place, children. The male and the female gamete, when it comes, the one set of DNA, another set of DNA combines together, you get genetic variation. Over here, there is no genetic variation. There is only a single egg cell that is forming the zygote. Single. So this particular whatever plant is being formed, okay, whatever seed is being formed will exactly look like this particular plant. Okay, agree? So I say that this process, even though it's not asexual reproduction, but it is like asexual reproduction. It is like asexual reproduction. Fine. And hence, whatever things that are got will be known as clones. They will be known as clones. So apomixis is the production of seeds from unfertilized ovule. It is true. You are producing seed from an unfertilized ovule. It is not getting fertilized. Now, embryos produced from apometic seeds are not generally identical to the parent plant. Is it like that? Aren't they identical to the parent plant? They are identical, okay, and here it is written they are not. So basically statement 1 is true and statement 2 is false. So this is correct, okay. Works. And now one more thing, since we have already discussed this, I also want to tell one more thing. There is one more type of apomixis given in your NCERT, where what is happening, you remember nucellus was there, remember nucellus was there, and this nucellus was the one that is helping in the formation of embryo sac. So whole thing transforms into an embryo sac and everything. But sometimes what happens is that, children, my dear little champions, some nucellus cells, they just stay there. Some nucellus cells, they just stay there towards the edges. Okay, and what happens basically, this nucellus cells, suppose this is the embryo sac that is being formed. This nucleus cell slowly divides itself and gets inside this embryo sac. Okay. So over here I am saying the nucellus cells which were there, they are getting inside, and later when they get inside, they will be forming embryo. How, when, what is not given in your 12th grade NCERT textbook, but yes, it is happening. Okay, so the nucellus cells getting inside the the sac will form an embryo. So over here, this, so multiple embryos will be there. So I call this condition as polyembryony. I call this condition as polyembryony. And where do I see this particular thing? I see this in mango and citrus fruits. I see this in mango and citrus fruits, children. Works. So I also wanted to tell that so that you don't get confused anytime if we see another question. Done with this. Okay, now we will try to make it a bit fast because I see 36 minutes are already over, right? So over here, okay, chalo. Select the correct sequence of events in microsporogenesis. So microsporogenesis, micro is for male, megasporogenesis, megaspore is for female, okay? Micro is for male. So microsporogenesis, as in the formation of microspores, what is the correct sequence of it? So basically again, over here, just like how we had nucellus, no? Similarly, you have so many cells coming together, okay? So many cells coming together, you call these cells, a group of cells, as tissue, right? I will give it a name, it is known as porogenous tissue. In the case of nucellus, only one cell was saying that I am the megaspore mother cell. Over here, all of them, they are saying that I am microspore mother cell. So basically, all of them, they are going to act as microspore mother cells. And you know what they are going to do. They are going to undergo meiosis. So this cell will undergo meiosis. This is the first phase where you had 2n, now it is n and n. In the first phase itself, it is reducing the number of chromosome. Again, this will also divide. This is the second phase of meiosis. You will get n, you will get n. So you have 4 number of gametes over there. Okay, now over not 4 number of gametes, please do not get confused. So, microspore mother cell, it underwent meiosis and now you have 1, 2, 3, 4. What are these 4? They are microspores, children. And since they are 4 in number, I call them microspore tetrad. I call them microspore tetrad. So in the case of megaspore, what was happening? 3 of them was getting degenerated, only 1 was forming the embryo sac. Here all of them, they will be known as microspore. They will be known as microspore, and this microspore, all of them, uniquely they will be forming pollen grains. So this will be one pollen grain, this will be another, this will be another, this will be another. So all of them will be having pollen grains. So just like how it was forming embryo sac there, over here it is forming the pollen grains over here. Okay, and now in the embryo sac, what was happening, children? The female gamete was formed. So I'll write embryo sac, since it is giving rise to the female gamete, I can call this embryo sac as female gametophyte. What do I call it? I call it, okay, I should write it properly. I call this as female gametophyte. Since it is the place where it is the one that will be giving rise to the female gametes. Over here, the pollen grains will be the ones that will be giving rise to the male gametes. They are not the male gametes, children. Please do not get confused with that. They will be giving rise to male gametes, which is why pollen grains are known as the male gametophyte. The male gametophyte. So over here, the correct sequence will be what? Sporogenous tissue will be forming microspore mother cell. Yes, microspore mother cell will be having a microspore tetrad, and each one among them of the tetrad, you call them what? Microspores. So this is the correct answer. I didn't have to go for the next options also. So if you remember this thing, it will be really, really easy for you. I know I might be explaining it too much, but I am explaining it to you because I want you to understand things. If I just wanted to randomly tell you answers, I can do that possibly, right? But I don't want that. I want you to understand things and learn. Okay. Now, sperm produced by the seminiferous tubules are transported through accessory ducts. Which duct should be tied and cut for male sterilization? So, ducts, okay. In the case of males, you have testis, that is the place where it is known as the primary sex organ. Okay, testis is known as the primary sex organ because this is the place where the male sperms and male hormones are being made. Okay, now this male sperms and hormones have to be carried also. So these are carried by accessory ducts. Okay, so you have testis. After the testis, you have accessory ducts which are responsible for carrying the sperm. Now there will be someone who will be waiting for the sperm to come so that they can give the secretions which will make the sperm be able to move more or be safe inside the female genital tract later on. So these things which gives out their secretion, you call them accessory glands. Okay, and lastly the sperm has to move out of the male body, and this organ is known as external genitalia, as an external genital organ, and this is the penis, children. And now you are asked, sperms produced by the seminiferous tubules. So seminiferous tubules, so you understand one thing, that sperm is getting produced inside something that is known as seminiferous tubules. So you have to understand that this is the, suppose the testis, okay. Inside the testis you have so many things like this which are known as testicular lobules. Okay, TL, team lead, you can remember like that. Now inside the testicular lobules you have so many, 1, 2, 3 seminiferous tubules. What do you have, children? 1, 2, 3 seminiferous tubules. And if I zoom this in, if I zoom this seminiferous tubule in, so this is one seminiferous tubule, this is another seminiferous tubule, and there is a space in between. This space is known as interstitial space. Interstitial space. So I am saying inside the seminiferous tubule, the sperms will be formed. The sperms will be formed from the sperm mother cell. Okay. in between the seminiferous tubule, I said interstitial space is there. Over here also you have special type of cells, you call them Leydig cells. What do you call them? Leydig cells. And what do Leydig cells do, children? Leydig cells are responsible for making the male hormones. So my two condition of the testis is done. One is to form sperm, it is done. The other is to form the male hormones, that is the androgens. Male hormones, what do you call them? You call them androgens. That is also done. Now the sperm which is made over here has to be transported. So the first guy who comes into play is, the first duct that comes into play is what? Rete testis. Rete testis. Fine. Rete testis comes here and forms something like this. Okay. This is known as vasa efferentia. So rete testis opens into something that is known as vasa efferentia. E for exit, you can remember like that. Okay, vasa efferentia. Now vasa efferentia will later say, I want to move out of testis, I don't want to stay here, I am carrying the sperms. So basically that moves out of the testis and forms something like this. It is very coiled structure. If I open this coil, it will be around 6 meters long. This very coiled structure you call this as epididymis. What do you call this? Epididymis. Remember, I am talking about the accessory ducts now. So this is the first duct, this is the second duct, this is the third duct. Okay, and epididymis, now this is the place where the sperms and everything are getting matured. The sperm basically as of now didn't move until now. It was because of the fluid that it was moving, but until now it was not gaining the proper structure. Okay, this is where it gains the proper structure, it is now mature and say that now I can go out. So basically after maturation and everything, the sperm goes out above to the urinary bladder region because it has to join to the external genitalia through which it has to go out. So this particular duct, this is the last duct that we are having over here, the accessory ducts, this is known as vas deferens. This is known as vas deferens, my dear children. So this is the fourth, this is the third, this is the which carries everything to the external genitalia. Now sperms produced by the seminiferous tubules are transported through the accessory ducts. Which duct should be tied and cut for male sterilization? So in the case of sterilization, as in you do not want the male to basically give out the sperm during ejaculation, such that pregnancy would not occur. Okay, so this process is known as sterilization procedure, where you have to cut a part of the accessory duct so that the sperm would not be going. So which part will you cut? Obviously cutting this won't help me. Okay, so this is the one that carries everything. I can easily cut this off and tie it off. So is it a problem, ma'am? No, no problem. Sperms will still be produced, but the sperm is not getting transported. Sperm will not be getting transported. Okay, outside. So what will be there then during a sexual process? What is the thing that will be ejaculated over there? It will be just the fluids which are given by the accessory glands. Accessory glands still does not know that the sperm has not come. So basically they give out the secretions, and this secretion is the one that is being released during sexual ejaculation. For the people who underwent this particular procedure is known as vasectomy, children. What do you call it? Vasectomy. Because you are cutting the vas deferens and you are tying it up. So sperm is still being produced in their body, but is not transported out, and it is not found in the semen. It's not found in the semen, as in the product that is being released from the external genitalia. In the external genitalia, semen will be released, but this semen will not have sperm in it. It will be just the fluid. That is what? Seminal plasma, which will be released by these guys. Seminal plasma is the secretions that are released by accessory glands. So basically that secretions will be there. Cool. So answer will be A. Works? Going ahead. In which of the following plants are both male and the female flowers born on the same plant? Okay. So when both male and the female plants are born on the same plant, you call them as monoecious. What do you call them? Monoecious. Where both male and female plant is there. Okay, now it can be the condition where the flowers are having both the female and the male parts, that is the the pistil and the stamen, or it can be a case where the flowers One flower is having the male, one flower is having the female, and the other flower is basically having the male. This can also be the condition. So either it can be this condition where it has both, or it can be this. Anyways, both male and the female plant part is there in the same plant. This condition is known as monoecious. Okay. Now, there is another condition that is known as dioecious, where you will be having a plant which will be just having the female parts, and there will be another plant which will be just having the male parts. So this condition where both the sexes are in different plants, both male and female in different plants, you call this as dioecious condition. Now, first question is, which plants are, in which of the following plants are both female and the male flowers born on the same plant? That is the first question. So over here, dioecious plants, they do not have. Papaya is dioecious, date palm is dioecious, spinach is dioecious. 3 of them are dioecious. I didn't even have to go ahead for the next part of the question, but still we have to go ahead with the next part of the question. Okay. And the mode of pollination can be geitonogamy or xenogamy. What do you mean by geitonogamy? Geitonogamy is when in a single plant, from the male part, things are going on the female plant. It's not different plants, it's the same plant. Okay. Geitonogamy is when everything is happening in the same plant, from the male part to the female part, or from the male part to the female part. Okay. If this is the condition, this is not geitonogamy. Okay. When, I don't want to confuse you, but I'm telling you, for example, both male and female is there in the same flower itself. In this case, it can just directly go here. This process is known as autogamy, where it's same plant, same flower. But in the case of geitonogamy, what is happening? Same plant is there, but flowers, if you see, they have to be different. Same plant, different flowers. That is known as geitonogamy. Okay. Here, geitonogamy, can it happen? Do you see male parts over here? From here to here, does anything go? Nothing will be going. So geitonogamy won't be found in dioecious plants. Now talking about xenogamy. Okay. Now talking about xenogamy. What do you mean by xenogamy? Xenogamy is when from one plant to another plant things are being going. From one plant to another plant. So this is the monoecious plant. Similarly, if there is another monoecious plant, so from here to here, obviously things can go. Yeah. So it is a different. Xenogamy is altogether different, where you are talking about different plants itself. It's not the same plant. It's different plant. So if you have 2 plants like this, so obviously from here to here things can go. Similarly, from here to here also things can easily go. So xenogamy is possible for both dioecious and monoecious. When you talk about monoecious plants, what all are possible, you have to see that. Okay, what all are possible for this particular monoecious plant? Geitonogamy is possible, xenogamy is definitely possible, and also autogamy is possible if the condition is like this. If the condition is not like this, then in monoecious plants also this cannot happen. Okay, agree? So as of now, we just have to see where both male and the female flowers are there on the same plant, and also the mode of pollination can be either geitonogamy or xenogamy. Geitonogamy or xenogamy, both is happening in the case of monoecious plants, and the answer will be maize, children. And you have to remember the names. There is no other way that you can know this answer. Papaya, date palm, spinach, all of them basically I remember like this. Papaya P, date palm has also P. Okay, and then S you can. Popeye the Sailor Man, you know the Popeye the Sailor Man. Do you have you watched the cartoon? So in Popeye the Sailor Man, he used to have spinach. That's how my parents used to give me spinach. Okay, saying that I will also become like Popeye the Sailor Man. So similarly, Popeye the Sailor Man over here is having spinach, and they are all dioecious children. They all are dioecious. They are unique in their own ways. Yeah. So this is the answer for this. Works. Are you understanding what I'm trying to teach you? Or is it just me blabbering around? So which one of the following hormones is secreted by human placenta that helps in the maintenance of pregnancy? Placenta basically is a thing that will be nourishing the embryo. Okay, so you know sperm, egg fuses, you have zygote. Zygote forms the embryo, and this embryo goes and stays in the in the uterus where the placenta is formed, and this placenta will be giving help or helping in the nourishment of this embryo that is being formed. So placenta basically is responsible for giving 4 hormones. One is human chorionic gonadotrophin, chorionic gonadotrophin. Okay, another is human placental lactogen. Okay, other one is estrogen and progesterone. So estrogen and progesterone is normally also released, but we say placenta releases 4 hormones. This also is there. Okay, now over here relaxin is given. Relaxin is released by ovary children, it is not released by them. Oxytocin is basically released by pituitary glands, even though it is formed in hypothalamus, but it's the pituitary that releases this. So you have this and this. So which one of the following hormones is secreted by human placenta that helps in the maintenance of pregnancy? So you know placenta is releasing this and this, but there the question is that who is responsible for maintenance of pregnancy? That is another thing. So maintenance of pregnancy, for that you have to maintain the whole uterus probably just like that. You have to make sure that the endometrium is layered just like that. So basically for doing that you need human chorionic gonadotropin. How is that possible, ma'am? HCG. How does that play an important role over here? So basically I'll tell you a small story. Okay, so ovulation happens. You know, ovulation happens and the Graafian follicle which is there, which we studied, it ruptures and the secondary oocyte is being released. So when the Graafian follicle ruptures, the Graafian follicle converts into something that is known as corpus luteum. Corpus luteum. So whatever thing is left behind when the Graafian follicle releases that thing is now known as corpus luteum. Okay, and this is being caused because LH has been increased too much, which is why ovulation has happened and everything. So LH is now in too much amount, FSH is also there, which is why the corpus luteum is now being formed. The corpus luteum, basically the remains of the Graafian follicle, will release something which is known as progesterone and also estrogen. Do you know what do? They basically, especially progesterone, they help in maintaining the endometrium. Maintaining the endometrium. Okay, why is this happening? So you only think, use your common sense over here. Okay, the egg is being released. Now obviously the body wants the egg to get fertilized. Now if by chance the fertilization has taken place, endometrium should always be there, because it has to. Endometrium will be the one that will provide the initial nourishment before the placenta is being formed, right? So endometrium has to be formed. So that is why it thinks that okay, we have to maintain corpus luteum. Corpus luteum will be releasing progesterone, estrogen, and this thing, whole thing will be happening just like that. Endometrium will be maintained. Now after a point of time when the body sees that there is nothing, okay, no fertilization happening, what will happen? Basically, whenever the progesterone is being made, you have to remember this progesterone says to the pardada of LH and FSH, that is the, what do you call this? I forgot. Human gonadotropic hormones. Okay, so basically it tells the hypothalamic hormones. Okay, I'm just writing hypothalamic hormones over here. Says the hypothalamus that you have to make your secretions less. Now we do not want your secretions. So this secretions affects the secretions of LH and FH, because hypothalamus is the one that gives commands to pituitary, and pituitary is the one that gives rise to both of these guys. So hypothalamus, when this is being stopped, obviously this says the pituitary, you have to stop your secretion of LH and FSH. Now you only think, if LH stops, corpus luteum stops. If corpus luteum is not there, progesterone is not there. If progesterone is not there, endometrium is not there. Isn't it doing a very wrong thing? Yes. So someone has to come and stop and say, no, progesterone, you do not have to do this. We have to maintain the endometrium because pregnancy has took place. Fertilization has taken place. Now we have to maintain the endometrium. So who is that person? It's basically the human chorionic gonadotrophin. Okay, human chorionic gonadotrophin comes to play and helps in the maintenance of corpus luteum. Instead of saying progesterone is now going to say to LH and LH, do whatever you want to do, no problem. I don't want LH now because there is someone else who will be maintaining the corpus luteum for me. It's not the LH who will be maintaining. And this is human chorionic gonadotropin. It will maintain corpus luteum. If it maintains corpus luteum, progesterone will be there. If progesterone is there, endometrium is being maintained. Okay. So which is why for pregnancy tests also, in the urine, the sample of HCG is being tested. If HCG is there, that means pregnancy is being there, which is why that coloration happens in the kit. Okay, so it is human chorionic gonadotropin which is responsible for maintenance of pregnancy. Even the placenta is releasing this also, but this is not responsible for maintenance of pregnancy. For maintenance of pregnancy, you need endometrium initially. So for that, you need this. Works? Are you getting happy? We are understanding different things over here. Yeah. Now the periodic abstinence by a couple for family planning should be. What do you mean by periodic abstinence? As in, you are absent in a period. Okay, so basically this is a natural contraceptive method where you are relying on your body and yourself for contraception. At this point of time, periodic abstinence means you are absent. Basically, you are not doing sexual activity. No sexual activity for a period. What period should be the best period for not doing sexual activity? The most fertile period, right? You should avoid having sexual activity during the most fertile period, children. And what is the most fertile period? On the 14th day of the cycle. You know the cycle is of 28 days. Yeah. On the 14th day of the cycle, the ovulation takes place, and that's when the graphene follicle ruptures and gives out everything. So for some women, what will happen? This could be shifted to this side. This could also be shifted to that side. So we cannot expect anything over here. Ovulation can happen on this day also, on this day also. So better not do sexual activity during these days. So what is the safest zone? You say 10 to 17 days. Sexual activity should not be done from 10 to 17 days of the menstrual cycle, such that you can be safe. So this fertile period, this is the fertile period. So this is known as periodic abstinence, and this is the answer, my dear children. Works? Now in humans, the secondary oocyte completes meiotic division when? I told you, secondary oocyte which was there over here. Remember? Where is secondary oocyte, children? Secondary oocyte which is stuck in the graphene follicle. Okay, it gets released when ovulation happens. Now, when fertilization happens, no, where is it? Yeah. Now, when fertilization happens, at this point of time, the zona pellucida is there. You know, remember? And then we have our little cell, that is the secondary oocyte is also there. At this point of time, when the sperm comes and attaches over here and releases its nucleus, at this point of time, the secondary oocyte will now divide into two cells. One is this cell and this cell. This cell will go die off. This is the second polar body. And this, my dear children, is the ovum. It finally has completed the division. It was stuck in metaphase 2. Now it has completed division and now it forms ovum. This is secondary oocyte. Okay. So, in humans, the secondary oocyte completes meiotic division when it gets implanted into uterine endometrium. No, it is released from the mature graphene follicle. At that point itself, it completes. No, it is penetrated by the sperm cell. Yes, acrosomal enzymes break down the zona pellucida. No, children, it is penetrated by the sperm cell at that point. Okay, acrosomal enzymes, what do you mean by that? Basically the sperm head, no, the sperm head, it has a nucleus over here. At the top of it, it has a crown which is known as acrosome. This acrosome has the enzymes which will be breaking down this particular layer of zona pellucida such that it can go reach the secondary oocyte. Okay, so acrosomal enzymes means enzymes which is there in the acrosome. So when acrosome tries to break the zona pellucida down, that's not when basically this thing happens. It's when the sperm cell is penetrated completely over here inside the secondary oocyte that this happens. Cool? Now these type of questions you find in NEET also. So I was thinking the level of questions coming in 12th grade now. Don't be panic, but yes, satark raho. We have to be prepared, right? So yes, the functional megaspore of an angiosperm develops into the functional megaspore. Remember, I told in the case of megaspore mother cell, it was dividing its cells, right? It was dividing, and now you had two cells, and then 2 will give rise to 1, 2, 3, 4 cells. 3 of them, they get degenerated. 1 will be there functional. This is the functional megaspore. Remember, what was it getting formed into? It was formed into embryo sac, children. Okay, this embryo sac later will divide and give the female gametes. This is the 10th question. Now, reasons for human population explosion are given below. Okay, human population. Now you have to think human population is increasing. Human population explosion as in it is increasing. Now the reasons: rapid decline in maternal mortality rate. Mortality means death rate. So rapid decline. If there is a decline in maternal death rate, that is mother's death rate, obviously if people are dying less, then the population will be more. So I am writing more population MP. Rapid decline in infant mortality rate. Again, there is a decline in infant mortality rate. People are dying less. Again, children, infant are dying less. So obviously again, population will be more. Now, rapid increase in death rate. If people are dying more, if the mortality rate is going high, will it cause population explosion? No. So this is less population. It is increasing the death rate. People are dying. So obviously there will be less population. Okay. Now increase in the number of people in reproducible age. If people are there, more number of people are there in the reproducible age, then obviously they will reproduce and give rise to more people again. So this will also give rise to more population. And you are asked reason for human population explosion. So this and this is the reason. 1, 2 and 4. Let's see. 1, 2 and 3 only. 1, 2 and 4 only. This is the answer. Easy question, no? This is easy question. Going ahead. This is the hormone that is secreted by the ovary in the later phase of pregnancy. I told you there is one hormone that is being released. This is known as relaxin. Why it is being released? You hear what happened. Relaxin basically, which is released by the ovary. Okay, it says after lots of stress, now you have to relax. It says to the pelvic ligaments that please relax. In that way only the baby can be moving out. The expulsion of the baby can take place. That is why it is asking to all the muscles and everything to relax slowly. Relax as in they will open up. Okay, so what will happen? The baby can be moving out easily. So relaxin is the one that is being released by the ovary. Always remember that. Okay, going ahead. This is the type of question that I was saying. Fill in the blanks. So they will give you a question and they will be giving you options like this also. Not for each question, they will be giving 5 set of questions, and for all of them they will be giving you options. So the interstitial space in the seminiferous tubules consists of immunologically what? So I told you about interstitial space. Remember, there is seminiferous tubule, one seminiferous tubule, there is another seminiferous tubule. There is interstitial space where you find Leydig cells, and Leydig cells are responsible for the male hormones, that is androgens. And if you look for Leydig cells, you cannot find them over here. So what will it be? The next type of cells that are found over here are the white blood cells, the WBCs, which are there to kill any type of pathogens that are going to come and attack us, or basically the cells. Okay, so immunologically, the interstitial space in the seminiferous tubules consists of immunologically competent cells, the one that can compete and fight. So this is the answer, my dear children. Okay, easy. Next, statutory ban on dash is required to check increasing female feticide. What do you mean by feticide? The killing of people. Okay, so female feticide is in killing of female people. So there was a ban done on something, okay, which was this particular process, X process was there, which was required for understanding the sex of the child, and this particular process was banned. What was this particular process? It was known as amniocentesis. Amniocentesis. What exactly happens in amniocentesis, children? So you know there is a baby. The baby basically is surrounded by a fluid. This is known as amniotic fluid. Amniotic fluid. Okay. Now what happens is that some cells of this baby will be shedding down in the amniotic fluid, and when people take it out with the syringe, the cells will also come along. And the cell's nucleus, if they check, they can see the number of chromosomes and everything that is there, but they look for something that is known as bar bodies. As in, if it's a female, you know there's an XX chromosome, and it's a male, it's XY chromosome. In the case of females, one X becomes deactivated and it will occur in the form of one small dot like this in the nucleus. In the nucleus you have the chromosomes, right? So in the nucleus you will be having a small dot over here. That means this is a female child. Whereas in the case of males, there is only one X, so that there is nothing to get deactivated. It would not be having the bar body. So the ones that will be having the bar body will be understood that, okay, this particular child is what? A female child. So amniocentesis was a thing that was banned. Cool? Works. 15th, make the uterus uterus unsuitable for implantation and the cervix hostile to sperm. What is that? Saccharomyces cerevisiae, that is yeast, adaptive radiation, hormone releasing IUDs, RNA interference. Basically it is hormone releasing IUDs. Okay, hormone releasing IUDs, examples are Progestasert, LNG 20. Basically all of them, they are hormone releasing. They release the hormone, that is progesterone, and you have studied that progesterone is the one that maintains the endometrium. But progesterone that is released in our body is released like that. But in the case of synthetic hormones, that is Progestasert, okay, in this case it is slow release of progesterone such that instead of making the endometrium thickened, it is making the endometrium thin. Now, if endometrium is thin, do you think the embryo can come and get implanted and the pregnancy can take place? No. That is why it won't take place. And also one more thing, what happens is that if you see the female body, if this is the uterus, this is the fallopian tube and everything, there is an area like this. This is known as vagina. And over here, there is an area like this. Okay, this area is known as the cervix. What happens is that when you are having hormone releasing intrauterine devices, this is one type of contraceptive method, intrauterine devices, okay, and this one is hormone releasing. What happens is that it forms a plug around here. It helps in the formation of a mucus plug around here such that the sperm would not be able to get inside. So it is making the cervix hostile to sperms. Okay, then second thing, it is making uterus unsuitable for implantation. By any chance that the sperm has come in, then also it is making the uterus unsuitable for implantation. This is hormone releasing IUDs. Apart from that, you have different IUDs. You have copper releasing IUDs, which will be releasing copper, and the copper will be like pulling on the tail of the sperm, does not allow the movement of the sperm around in the uterus. That is copper releasing IUDs. Then there is one more thing, that is normal IUDs, okay, not medicated IUDs, lipase loop. What happens in them is that it's foreign particle. It's what? Something foreign. And when something foreign comes in our body, what will our body do? It will fight against it. And while fighting against it, obviously it makes this particular area very unsuitable for implantation or anything. That's how the IUDs work. Okay, so this is the answer for this question. Cool. And now we are coming to the short answer type question, 2-marker question. Let's begin the battle. Yes. Chalo. Identify A, B, C and D in the table given below. What is pericarp? What do you mean by pericarp? So, if this is the ovary, this is the ovule. Ovule will be the seed. Seed will be having the seed coat and everything. The ovary which is there, it will be becoming a fruit. And fruit also has covering. This covering is known as, children, the covering of fruit, or basically the covering of the outer wall. I'll write the outer wall of fruit is known as pericarp. So if you see, if you see a mango, people who are attending NEET classes from me know the answer for the same. So basically over here, if this is a mango, this is the outermost covering, you might think. No, it's not like that. So pericarp is again divided into other things. So this is the outermost thing, the yellow color thing that you see, it is known as epicarp. Then something that we eat, that is also a part of covering, it is known as mesocarp. Okay, then you have gutli, right, the tough layer that is inside, it is known as endocarp. Now inside the endocarp, you will be having this particular thing, that is the seed. Seed will have its own covering as well. Okay. Understood? So these three layers coming together, you call it as pericarp. So in some, the pericarp is not like this, it is not fleshy like this, it will be just dry, that is different. But in some, it may persist. So it will be what? Over here, it will be outer wall of the fruit. So I can write over here, outer wall of the fruit. Okay. Now cotyledon in seed of grass family. So grass family. Grass is an example of monocotyledons. They do not have two cotyledon. They just have one cotyledon. What is cotyledon, children? So basically what happens is that in the case of plants, they have cotyledons which will be providing nourishment to the embryo. So in the seed, I said that the egg cell, okay, just a second, the egg cell which was there, it will undergo fertilization and will form the zygote. Agree? And zygote will later form the embryo. Agree? Remember the two nuclei that were over here? This will fuse with another male gamete. So when the pollen grain comes, they come with two male gamete. One male gamete fuses with the egg cell, other male gamete fuses with the two polar nuclei. Okay, and this fusion results in something that is known as endosperm. This will result in the formation of something that is known as endosperm. It is known as primary endosperm cell, which will later form endosperm, the whole endosperm. Okay, now this endosperm is the one that usually provides nourishment to the embryo. But in some plants, what happens is that it says that it's okay. Embryo says that I want to nourish myself in my own ways. So whatever things are there in you, just give it to me. Whatever nourishment tools are there in me, just give it to me. So I have my own things, and that things are known as cotyledons. So you might have seen green pea and everything, any pea and all, when you break it out, you get two pieces out of it. So these are cotyledons. These are cotyledons, and inside that you might see something is there, which is the embryo. So basically what happens is that this cotyledon provides nourishment to the embryo, and the embryo grows, and that's when you have the root, shoot and everything coming along. Okay, so cotyledon in the case of grass family, it's not like this. These are dicotyledon, but there are something else which are known as monocots. In the case of grass family, they are known as monocots. Okay, what happens? They do not have two cotyledons as you see over here. They just have one cotyledon. Okay, they just have one cotyledon, and this one cotyledon has a specific name. It is known as scutellum. It is known as scutellum. So you have to write it over here. Scutellum. When you have two cotyledons, you call them two cotyledons. When you have just one, you have a specific name for it. You call it scutellum. Apart from grass, there is wheat, there is rice, all of them, they are monocots. Okay. Now, embryonal axis. What is embryonal axis? Basically, I said, so what happens if two cotyledons are there? There will be an axis like this. Okay. These two cotyledons and this particular thing, which will be having a plumule at one end, which will be giving rise to the future shoot, and there will be having a radicle at another end, which will be giving rise to the future root. This particular thing from here to here, you call this as embryonal axis. Embryonal axis. This is for dicots where you have two cotyledons. Now over here for monocots, from one side everything will come. So this will be the radicle, this will be the plumule. Okay, they do not have two cotyledons on either side, they just have one. Okay, this axis that you see over here is the embryonal axis. What does it do? Gives rise to shoot and root. Works? Now, dash is the remains of nucellus in a seed. Okay, what do you call the remains of nucellus in a seed? So I said the nucellus is usually occupied. Remember nucellus? In the ovule you had nucellus and one becomes the megaspore mother cell and everything. So sometimes what happens is that over here, as I said, nucellus remains. Nucellus, it remains. Okay, and this nucellus will also be providing nourishment to the growing embryo which is over there. So now this nucellus has a specific name. You call this as perisperm. Do not get confused with pericarp. It is perisperm. So example of fruits that have perisperm is basically beet. Okay. Yeah, beet is an example of this. You can remember like that. Beet is an example of this. And pepper, I think. I don't remember exactly. Okay. Works. So that is identify A, B, C, D. Done. This is how you get 2 marks. So for 2 marks, you need to know all this thing. You see how enlarged your knowledge should be. So you should be trying for that. You should be learning you should be actually, I'm so sorry for confusing you guys because I'm so tired of talking a lot. So over here, yeah, the easiest thing that I wanted to tell is, do not think that is impossible by you. It is very much possible by you. If you sit and you just go through your NCERT in a very easy manner, you will understand each and everything of it. And now if you're not understanding, I have my videos for NEET over there in the YouTube channel itself. You can watch those videos and keep in 2x speed and watch it. No problem. You will understand. If you have read your NCERT and then come, you will understand it. There might be certain information which is extra for you. Skip that portion and go on with NCERT and just see what you have to study. But you have to study that, children. Okay, you have to study your NCERT very nicely. Then only you will be able to give answers. Now, write the basic steps followed in the assisted reproductive technologies, that is ART program, to help childless couples. Why is it also known as a test tube program? Okay. So assisted reproductive technology, assisted as in something is being assisted, you are not doing it very naturally, okay? When it is happening naturally, you do not need assisted reproductive technologies. So in assisted reproductive technologies comes IVF and everything, in vitro fertilization, everything. Why it is also known as test tube baby program? Because this is happening in the lab, children. This is happening in the lab, which is why it is known as test tube baby program. Fine. Now, how will you frame the answer for the same? It's a 2-marker question. It's a 2-marker question. This is the basic framework that you need to have. Okay. You can obviously club the points together. You can write hormone stimulation, egg retrieval and everything together and then write the answers, but you should be having the basic things there. So first thing that you do is basically, you know that the egg has to be released. So for egg being released, you do not need just one egg. One egg, one sperm, obviously there are high chances of failure. So we need to take a lot of egg, and for that a lot of egg has to be matured. Basically lots of follicles have to be matured and releasing the ovum, releasing the secondary oocyte. Basically you have to have the follicles around, okay? Normally one follicle matures and it gives the secondary oocyte every month, right? So over here we need more, so that's why we need hormonal stimulation. So that is the first step, where we are stimulating with the help of hormones so that multiple eggs are released. Now egg retrieval is done, mature eggs are being taken from the ovary. Sperm collection is done, males are asked to give the sperm, okay? Now fertilization is done where? In the lab itself, okay? Later of that, fertilization is done and then embryo culture is done. This particular fertilized egg is now grown in the laboratory condition, and then later when the embryo is fine, the selected embryos are transferred into what? The woman's uterus, or basically the fallopian tube, depending on what? Depending on the number of cells or the blastomeres. Okay, this basic thing can you write? You don't, if you want, you can give the example of GIFT and everything, that is zygote intrafallopian transfer, or gamete. When you are transferring gametes directly, you are not waiting for zygote and everything. For example, a female is not able to produce ovum. So other female is giving that ovum, you just have to put that ovum in the other female's body. This is known as gamete intrafallopian transfer, where you're transferring the gamete into the fallopian tube. Okay. Let's just like that things can happen, my dear children. So fertilization occurs outside the body in a lab, hence it is known as test tube baby program. Done, children. So this is how you have to frame. You can club these 3 points and then you can write the answer. Cool? Works for you? Now next, differentiate between gytonogamy and xenogamy. I already told you, when it is the same plant, okay, when it is the same plant but different flowers, you call this gytonogamy. Okay, when it is the same plant, I'll write it, it's visible. Same plant, different flowers, this is gytonogamy. Xenogamy is basically when there is different plants altogether. Different plants altogether. In that case, it is kind of different. Okay. Now, what will be much more, if you want 2-marker question, you have to put more points, right? What will the next difference? So here, don't you think genetically it will be the same? The product at the end will be genetically same because the same plant is being involved. So I am right, genetically similar to parents. Okay. Over here, what can I write? They are genetically different. So more amount of variations can I expect over here? This can be put as 3rd point. More amount of variations over here, no much variations will be found. Okay. Now, isn't it just like self-pollination because it's the same plant, but here it is acting as cross-pollination because it is different plants. So even though it is also coming in the same category, but this is basically acting like self-pollination only because it's the same plant that is being involved. So over here, these are the points that are given. Definition, you have to write a definition. Transfer of pollen grains from one flower to another flower of the same plant. Transfer of pollen grains from flower of one plant to another flower of another plant of the same species, okay, of the same species. Genetic diversity involves genetically similar gametes, so no new genetic variations, but over here they will promote genetic diversity. Functionally similar to self-pollination, even though it is not. Okay, why do I call it self-pollination? When same flowers are coming in contact, okay, flower parts are coming in contact, you call it self-pollination, right? So over here functionally it is happening even though it's not actually true. Okay, now true cross-pollination as it occurs between different plants. So gytonogamy and xenogamy both are coming under cross-pollination itself. So whenever it's between two flowers, you call it cross-pollination. Either the flowers can be on the same plant or on the different plants. But functionally, when I speak of gytonogamy, it is basically having the same genetics and everything. It is same acting as self-pollination only. So functionally, it is self-pollination, but actually it is cross. Okay, that is the point that is given over here and also in your NCERT. Next, do you think that apomixis can be compared to asexual reproduction? Justify your answer. State the benefits of apomixis to the farmers. So we studied about apomixis, which is why I taught you about apomixis, because I saw the questions over there. So this is a 2-marker question. Again, as I told, you can explain apomixis. Yes. Do you think that apomixis can be compared? Yes, I think that apomixis can be compared to asexual reproduction, because here production of seed is occurring without the fertilization. And if there is no fusion of gametes, children, if there is no fusion of gametes, obviously that progeny will be genetically identical to the parent, similar to asexual reproduction. Okay. Now here you don't have to explain the methods of apomixis, because it's a 2-marker. So over here 1 and 2 same. Okay. So from here also you can put down at least 2 points or at least 1 point. Okay. So how it is important for the farmers? You only think, I have a plant and this plant is perfect for me. This plant is disease resistant. This plant is everything that I want. If this plant is doing apomixis, I will get so many seeds, and the seeds will give me so many plants of the same nature. So uniformity in crops will be there. Okay, with the traits that I am liking, that is high yield and disease resistance. So that is one thing for the farmers. Okay. Second thing is stability in hybrid characters. So for example, what happens is that I have a character, but if sexual reproduction is happening, what will happen? My character, that character will also come together, and now you might have different characters. But I want this particular character itself. So I want stability in this hybrid character that I am already having. So for that, for obviously my seeds, the same seeds will be used. If that is used, then this particular character is preserved. There is no point of genetic variation. Genetic variation won't be occurring. Okay, so that is exactly that I have written over here. Apomixis preserve the hybrid character in successive generation, as no sexual reproduction is involved over here. Okay, so there is no chance of variation. Now, cost effective seed production. Obviously, I don't have to make them fuse, fertilize and everything. No, I have the seeds and I can use it. So this is cost effective as well. So among them you can write 2 points. That will be enough. Works? Now, name any 2 outbreeding devices that flowering plants have developed and explain how they help in encouraging cross-pollination. So, outbreeding devices, as in the plants are tired of inbreeding. What do you mean by inbreeding? Inbreeding is when, children, the plant is basically, for example, I have a plant, there is a flower like this. Okay. So over here, from here to here, things are getting transferred and everything is happening. Okay, everything is happening. Again and again and again and again, there is no chance of pollination, cross-pollination, nothing. Okay, or probably when things are like this, when you have both the female and the male gametes over here. Here there are high chances of self-pollination happening. Here there are high chances of self-pollination happening. So every time this pollen will come and land on the stigma and the same thing again happens, the plant will now not want this. And at one point of time, it might turn out to be infertile also. It might come to be sterile also. This process is known as inbreeding depression. Because of regular inbreeding that is happening, the plant itself is depressed. Okay. So now, the plant itself have brought out outbreeding devices. To not allow this to happen, the plant itself have given out outbreeding devices. Okay. And this outbreeding devices obviously will help encourage what? Cross-pollination. Cross-pollination as in not the cross-pollination like this, cross-pollination like this between one plant and another plant. Okay. So let's see the inbreeding devices that are given. One is self-incompatibility. This is a genetic mechanism that has been taken by the plant itself. Okay, it's a genetic makeup change that has been taken. Over here, what happens is that the pollen from the same plant or genetically similar plant is rejected by the stigma. So you know, if this is a stigma, if this is a stigma, okay, when the pollen comes and lands on it, if everything is good, the chemical reaction that is happening over here, it's good. That's only when this particular pollen can form the pollen tube. If this is not good, then pollen tube formation will not be happening. So now the plant itself has said no more same type of pollen or pollen from the same species. I don't want that. So obviously they won't be able to form the pollen tube. This is known as self-incompatibility. This is a genetic mechanism that has been done by the plant itself. Now maturation of stamen and pistil at different times. So just think, this is the flower. So over here you have the pistil, okay, and over here you have the stamen. This is maturing at a different time and this is maturing at another time. Obviously if it is come over here, lands on this, but this is not matured, then obviously the process won't be taking place. Or yeah, either way the process is not taking place. Or when this is mature, this is not mature, so there is no pollen to come and land on this. So obviously some other plant will give the other pollen grains, right? So maturation of stamen and pistil at different times, that is also one more thing. When the stamens mature first, okay, when the stamen, this thing is getting matured first, you call this protoandry. Okay, that is written right over here. Now when the pistil is maturing first, what do you call it? Protogyne. But over here you have to make sure both of them are maturing at different types. So these are the two outbreeding devices. There are much more given in your NCERT textbook as well. Then now we'll move on to the next question. The 21st question, in the figure given below, parts A and B show the level of hormones which influence the menstrual cycle. Okay, study the figure and answer the questions that follow up here. Name the organs and glands which secrete the hormones represented in the label parts A and B. So you have to tell the organs, the glands, everything that is being releasing all these hormones. So it is of 1 mark, so you will answer accordingly. What do you have? You have hypothalamus, which this is a gland that signals the next gland, pituitary, and you have to mention anterior pituitary, my dear children, which releases hormones LH, luteinizing hormone, and FSH, follicle stimulating hormone. Okay, so luteinizing hormone, I always mess up with the spelling, please check. Okay, and follicle stimulating hormone. LH and FSH being by this is mentioned in A. Second thing that you have to mention about estrogen and progesterone. Estrogen and progesterone is released by the ovary. So ovary releases estrogen and progesterone. This is B. Works? Okay. Now state the role of hormones secreted from part B on the uterus of the human female during the menstrual cycle. So part B, it is estrogen and progesterone, as you can see over here. When estrogen and progesterone is there, okay, so this is a phase where the endometrium lining has been fallen down. Okay, the menstruation has taken place. Now after this, slowly we want the endometrium line to form again. We want the endometrium line to form again, and which is why estrogen is being there. Okay, so what happens is that when the follicles, FSH is there, the follicles they generate, they mature, and the cells of the follicles, they will be releasing estrogen, and the amount of estrogen will go high. When the amount of estrogen, oh my god, just a second children, which is it? Yeah, when the amount of estrogen it goes high, just give me a second, I touch somewhere and everything is not visible. Okay, estrogen will make the endometrium lining which is shedded off to be formed again. Okay, it gets formed again. Now what will happen? The Graafian follicle which is there, it ruptures off. Why it ruptures off? Because the estrogen, as you see over here, as it peaks off, I think I am confusing you guys. So just a second, I'll just rub this off and I explain you once more. Okay, listen to me very carefully. Over here you have estrogen and progesterone. Assume that the menstrual cycle has taken place, the time where the periods have come, the blood and everything has flown down. Okay, we do not have the endometrium lining properly over there. Now we need to make the next layer of endometrium lining. Okay, so this point of time, what happens is that the follicles are generating, the follicles, they are maturing, and while they are maturing, the cells, the granular cells in them, they are going to release estrogen, just estrogen. So the amount of estrogen, as you see, it will slowly peak up. When the estrogen peaks up, what will happen? The endometrium line will also start forming, my dear children. Okay, but estrogen, when it reaches a peak, what will happen? The LH will also reach a peak and FSH will also suddenly increase. At this point of time, this particular thing, that is the Graafian follicle, it ruptures to give out the secondary oocyte. And the Graafian follicle which is there, it will now slowly transform into something that is known as what? Corpus luteum. Corpus luteum, I told you, what does it release? Corpus luteum releases progesterone. So now the progesterone, which was not there until now into the play, comes into play as it is being released, and its amount increases very much. Similarly, estrogen is also being released. So both of them are there, and now what will happen? Because of the release of progesterone and estrogen, this endometrium lining slowly moves to be very, very big. Okay. Now later on, the progesterone, as I said, will go tell the GnRH, gonadotropin releasing hormone, to stop their secretion. If it stops, the LH and FSH stop. If LH and FSH stop, this corpus luteum will be degraded, and now everything will fall down. Then the endometrium lining, the progesterone will not be there, estrogen will not be there, again everything falls off, the sheds off. Okay. So how do you write the answer? State the role of hormones secreted from part B. So we will write initially, okay, granulosa cells release estrogen, which forms the endometrium, initially, and when the estrogen peaks, LH surge occurs, and what do you have, children? You have ovulation. Because of this estrogen only, things are happening like that. Now the next thing, now comes the role of progesterone. So you have to write about that. This is the 2 points that you can write about them. Now about the role of progesterone, where can I write? Because if I use a second slide, everything will be gone. But I'll write over here. Okay, I'll write over here because this is not a very big answer. That is the role of estrogen. But when you talk about progesterone, what will happen? Now the graphene follicle will be transforming into corpus luteum, and the corpus luteum will be releasing what? Progesterone and estrogen, which will do what? Which will build the endometrium even more, or you can say it will, instead of build, you can write thicken the endometrium. Thicken the endometrium such that if fertilization has taken place, it can give rise, it can take care of the the progeny, okay, the embryo. That's all about the answer. That's how you have to frame the answer. So if you are not understanding things, you won't be able to get it. So it's the follicle cells that releases estrogen when the follicles, they mature, okay, and this estrogen maintains the endometrium line for some time. Then the follicles, when they give out secondary oocyte, and the graphene follicle becomes corpus luteum, that's when it releases progesterone and estrogen, which thickens even more the endometrium, even more thickens the endometrium layer. Fine, that's how it works. Now when the level goes down, again the endometrium also falls down. 22nd question. Suggest 3 simple principles through which we can prevent sexually transmitted diseases. Sexually transmitted diseases obviously will be transmitted during the sexual intercourse. Okay, so that means semen has a big role to play. All the genital fluids have a big role to play over here, and usually sexually transmitted diseases basically are transmitted through blood as everything as well. Okay, so basically what are the steps? First thing, use of condoms, that is the first step. Condom is acting as a physical barrier. It's the only physical barrier, or it's the only contraceptive method that prevents what? Sexual diseases. There is no other contraceptive method that prevents sexual diseases, and not all type of condoms, the male condoms. Okay, there are female condoms also, but the male condoms are the ones that prevents this, because in the case of male condoms, the penis is covered by the condom, so there is no chance for the fluid, anything, to come in contact with the female genital tract, which is that is the vagina. Okay. Now next thing, avoid multiple sexual partners. So multiple sexual partners, there are high chances that one partner could be infected and you could get the infection as well. So be sure of having just one sexual partner, and for your age, none. Okay. Next thing, take regular health checkup. Take regular health checkup so that if there is any sort of problems, probably you can prevent passing it on to another person, and that's also a step of prevention. So these are the 3 steps. It's given in your NCERT textbook. You have to learn it, you have to write it. Okay, works. Going ahead, draw a label diagram of transverse section of young anther. So your anther looks like this, children, and I'll explain it to you. Yeah, works. So over here, let's make. So this is your stamen, where this thing that you see over here is the filament, and this is the anther. Anther is bilobed. Anther is what? Bilobed. It has 2 lobes. So basically, if I cut the anther like this, I can see something like this. Okay. This is one lobe. This is another lobe. This is one theca. This is another theca. So I say that each lobe is dithecous. Each lobe is what, children? It is dithecous in nature. So over here, the innermost thing that you see over here, this is, this whole thing you call it as a microsporangium, because basically the cells that you see over here, it is the sporogenous tissue. And I told you each cell of the sporogenous tissue can form the microspores, right? Now, the sporogenous tissue is basically covered by a layer of next type of tissue that is over here. You call this as tapetum. You call this as tapetum, and tapetum is the only layer which provides nourishment to the growing cells, that is over here. Okay, it provides nourishment. Now, outside the tapetum, I'll start making from here now. So, over here, I'll have certain cells, that is the outermost cell, you call this as epidermis. Okay. Now, inside the epidermis, you have the next type of cell. You call this as endothecium. You call this as endothecium. And then you find over here, this particular layer that you see, this is the middle layer. So how many of them you have? 1, 2, 3, 4, and then inside you have the sporogenous tissue, which will be having the microspore mother cells, which will be forming the microspores. So epidermis, endothecium and middle layer, the 3 of them, they are responsible for what, children? They are responsible for protection and for dehiscence of anther. Okay, so when anther breaks open and everything, it is responsible for that. Tapetum is basically responsible for nourishment. And this is the structure that you have to remember. This is the structure of the young anther, transverse section of the young anther that you have to remember. This is given in your NCERT like this. Works? Going ahead. Draw LS of grass embryo diagram and label the following parts: scutellum, coleoptile. Okay, I told you. There is only, in the case of grass, it is a monocotyledon. It has just one cotyledon. So in your NCERT, the picture is given like this. I find it to be very confusing. So instead of that, let's make our own diagram. It should be understandable. It should have all the parts, right? So basically over here, I have the embryonal axis and I have the scutellum. This is the cotyledon that I am having. Okay, what is this, children? The scutellum. So, I am writing scutellum. I told you the endosperm is totally utilized in the case of dicot, which is why they have 2 cotyledons. In the case of them, they are just making one cotyledon, so they still have the endosperm still left. So, this is the endosperm that is being still left. Leftover endosperm. Okay. Now, this will be the plumule. This will be the radicle. And there is a root cap over here, so that the radicle does not get damaged. Okay. Now, we are done with coleoptile. Okay. Now, we are done with shoot apex. This is the shoot apex, my dear children. It forms the shoot. Okay. And root Radicle is there, so radicle I have mentioned, root cap also I have mentioned. Now coleorhiza and coleoptile is there. So over here they have also mentioned like this. How we can explain is that we can make things like this. This thing that you see over here, which covers the plumule, that is the coleoptile. Similarly, there will be something like this, which will be covering the root. This will be known as the coleorhiza. Works? Okay. So scutellum is mentioned. Scutellum is mentioned. That is the first thing. B, what you have to mention? Coleoptile. Okay. C, what you have to mention? The shoot apex. D, what you have to mention? The radicle. E, what you have to mention? The root cap. And F, coleorhiza. Works? And this is the picture that they have given. You can explain it their ways also. If you want to copy exact thing, you can do that also. But according to me, that does not make any sense. Okay. Now, parturition is induced by a complex neuroendocrine mechanism. What do you mean by parturition, children? Parturition is basically expelling out of the human baby after it's grown fully, completely. Now I say it's a neuroendocrine mechanism. That means basically my nervous system is involved, my brain and everything is involved. Apart from that, the hormonal system is also involved over here. That is what I mean by neuroendocrine mechanism. Okay, so parturition, I say it's like this. It is handled by both. How can I explain this? With the help of this. So endocrine signal, what happens? I'll explain it to you pehle, then I'll go ahead. I think that will be better. So basically, when the baby is in the womb, so over here, let's think the baby is there in the womb. I'm not making the proper diagram, but yeah. The fully grown embryo, whether male or female, they will be releasing something that is known as androgen. This androgen is taken to be converted into estrogen. It will slowly help in the formation of estrogen, and this estrogen basically will do one thing. So we have so many layers. Okay, we have so many layers in our uterus. The outermost layer is perimetrium. Then comes myometrium. And then comes endometrium, right? So this estrogen which is there over here, it increases the number of, I'm making over here, number of certain receptors on the myometrium. Increases the number of certain receptors. What are these receptors, children? Oxytocin receptors. So there's a hormone that is known as oxytocin. Its receptors are increased. So usually what happens is that when receptor and the hormone comes together, that's when the working starts. But over here, the hormone is being released, but there are no much receptors usually present. But at this point of time, when the fully grown embryo is now releasing androgens from them, and the androgens basically is helping in the formation of estrogen, and the estrogen says through myometrium that you have to increase the number of oxytocin receptors. What will happen? Basically, the oxytocin which is released by the posterior pituitary. So posterior pituitary under the effect of hypothalamus. So actually oxytocin is released by hypothalamus. This is stored in posterior pituitary, and when this release oxytocin, what will happen? This oxytocin will go bind over here, and when it binds, children, when the oxytocin comes and binds over here, this myometrium that you see over here will start contracting and relaxing. Will start contracting. Now something is getting contracted. Obviously, don't you think that this particular baby that is over here, fetus, will be moving down? Yes, it will be moving down. Now when it moves down, again it gives signals. Again it gives, there will be other stress signals over here. More oxytocin will be released, more receptors will get activated, and now it will go down like this. This is known as fetal ejection reflex. Okay, what do you call this? Fetal ejection, as in you're removing, ejection reflex, which is getting started off from the baby itself, in the fully grown fetus itself. And now over here you see there is a control of hormones, there is a control of hormones that you see over here, estrogen, oxytocin and everything, but there is a neural control as well, because this effect is being shown initially by hypothalamus. Hypothalamus has to get activated to give posterior pituitary this step or to say to the posterior pituitary that, hey, whatever oxytocin that you have stored over here, please, it's time to release now. Basically, it's always releasing, but still there are no receptors here there to attempt there to catch of this oxytocin, but now you have receptors also. So now the secretion increases tenfolds. So there is a combined effect of both of them. That's how things work. And now if you want the answer, how you have to frame the answer, you have to write like this. So estrogen towards the end of pregnancy, estrogen level rise, which stimulates the uterus to become more sensitive to the action of oxytocin. How? Because you are increasing the oxytocin, basically increases the synthesis of oxytocin receptors in the uterine muscles, preparing the uterus for stronger contraction. So instead of writing uterine muscles, you can write myometrium. Okay, and this is the first point that you can write. Okay, or you can write towards the end of pregnancy. This is the first point. This is the second point that you can write. Now, released by the posterior pituitary gland, oxytocin stimulates uterine contraction. It also aids in, okay, this will be the next step that you can write. This is the third point that you can write. Now, neural control, and that you can write the stretch of the cervix is detected by the sensory nerves that send signals to the brain. So, in the cervix there are sensory nerves, and obviously this nerves, when they get activated, they will send signals to the brain. The hypothalamus responds by stimulating the release of oxytocin, which promotes stronger uterine contraction. So, more amount of contraction will be there from that angle also. Okay, so you can write these points as well as these points. So, this is the endocrine signal, this is the neural signal that is getting. Okay, here also you can include the neural signal, but still, just like how it has been written, you can write it. Cool, works. And you can say towards the end of pregnancy, it's because of the fetal ejection reflex. Please write like that. Fetal ejection reflex, estrogen is released, and always write your answer in points, point to point. Works? Going ahead, draw a neatly labeled diagrammatic sectional view of male reproductive system. Okay, so the male reproductive system looks like this. How we can draw it? Let's see. Over here, initial thing, you have testis 1, and this is testis number 2. So I'm just making one testis. Okay, so over here we have the testicular lobules, my dear children. Okay, testicular lobules will be having seminiferous tubules. Agree? So make like that. Label everything. So I am here. Okay, I'll label also. This is seminiferous tubule, and this is testicular lobule. Okay, now from here, this thing that you see, it's known as rete testis. It is known as rete testis, and the rete testis basically are combining together over here. This will be what? Vasa efferentia. Vasa efferentia will be opening up. It's not mentioned properly over here in your NCERT, but still. Okay, the vasa efferentia, it is there. Actually speaking, I did a small mistake. Not a small mistake, very big mistake. Sorry children, in the other one also, please correct it. Okay, this is not vasa, so this is not tubuli recti, this is tubuli recti. That is something else that is not given in your NCERT. I'm so sorry. So this thing that you see over here, this is rete testis. Okay, this is rete testis, and the rete testis, they open up here like this. They try to exit, and this is known as vasa efferentia. Okay, so tubuli recti, there is one more thing that connects rete testis and the seminiferous tubule. It is known as tubuli recti. It's not given in your NCERT. Okay, now vasa efferentia is there. Vasa efferentia will come, it will coil up like this. And what do you call this structure as? Epididymis. Okay, what do you call this structure as? Epididymis. Now from epididymis, one thing goes like this. From here, slowly come like this. This is known as vas deferens. Okay, and vas deferens, as you can see over here, it coils over the urinary bladder. So I have to make the urinary bladder. So over here, I make the urinary bladder like this. Okay, and urinary bladder is having ureter. Okay, and so it will coil up and it will come like that in front. From back it will go and from front it will come back. Okay, now the thing is, at this point it will be releasing a secretion from a gland which is known as, where should I write? Here only I'll write, seminal vesicle. This is the first accessory gland. What do you call this? You call this as seminal vesicle. Okay, now basically this will now, both of them will be opening into something like this. This is the ejaculatory duct. Ejaculatory duct. So basically over here if I mention properly, so you remember you had the vas deferens, and this vas deferens is basically getting its secretions from the seminal vesicle as well, and now both of them, they will be opening into the what is this? Urethra, with the help of a common duct, and this common duct is known as ejaculatory duct. Okay, now one more thing you have to remember, basically there will be a gland over here that covers everything like this. This is the second gland that we have, and this is known as prostate gland. Prostate gland. So first gland is seminal vesicle, which is releasing the fructose and everything that provides energy. Second gland is prostate gland that provides calcium and everything, okay, and certain other enzymes. And then from here starts the external genitalia. So over here, external genitalia, it looks like this. Okay, you have the glans penis, and the urethra opens up here, and this will be known as the urethral meatus. This is known as the urethral meatus, my dear children. And over here there is bulb shaped gland, that is the third gland that we are having, and this gland is known as bulbourethral gland. Okay, so what you can do to make your picture really good, better than this, is that you can mention the glands in color in black pen. Okay, you can mention the glands in black pen and then other ones in blue pen and other ones in pencil, so you have a clear differentiation with that. Okay, and this whole external thing that you see over here, this is the penis. And if you want, you have to explain the foreskin as well, so please explain the foreskin there. Okay, so there is a glans penis and there is a foreskin over here, right over here. Works? Please don't put foreskin over here. This is the urethral meatus. That's how you have to draw, my dear little champions. Okay, so I'll explain. Over here, the seminiferous tubule releases sperm. You have the sperm travels to the vas deferens. It gets the secretion from the seminal vesicle, which gives what? Which gives this thing, fructose. That is the initial form of energy. Then prostate gland gives what? Calcium, which helps in the movement of sperm, and also helps in certain changes in the sperm, okay, which will be helping in fertilization. And then bulbourethral glands will help in the lubrication such that the sperms can easily travel through. Okay, and then it travels through. Over here you have the urethral meatus, through it moves out. You have the foreskin, and this enlarged portion is known as the glans penis. That's all. Works? Okay, and also remember one thing, in the case of men, as you can see over here, the urine and the sperm travels to the same place, and that common duct is known as urethra, and that common area is known as urethra. Works? Now, picture of a mature angiosperm embryo sac is given below, and answer the questions that follows. This is a 5-marker question. This was also a 5-marker question, so you have to properly draw a diagram and then only explain things. Okay. Now, which cells or nuclei of the embryo sac produce zygote and primary endosperm nucleus? So I already told you. So over here, okay, over here if things are mentioned like this and some handy stuffs are mentioned over here, just understand that these cells that you see over here are synergids. Okay, and this handy structure that you see over here is filiform apparatus. Why do you need filiform apparatus? Basically, when the pollen tube comes, the pollen has to come towards the egg cell, which is over here. This cell that you see over here, this is the egg cell. So the filiform apparatus from the synergid guides the male gamete towards the egg cell. Okay. So now egg cell is there. So this egg cell is going to help in the formation of zygote. That is the first thing. So which cells or nuclei? So zygote is the first thing. So it is being made by the egg cell. Okay. Now if you are asked about the primary endosperm nucleus, so I told you there are two gametes that comes in. First gamete goes and fuses with this. The next gamete goes and fuses with this. So these are not cells, children. These are just nuclei. They are just nuclei. You call them polar nuclei. So polar nuclei plus the second gamete will be making what? Will be making what? The primary endosperm nucleus. Will be making the primary endosperm nucleus. Okay. So basically this is the actual cell and these are the two nuclei. This whole thing that you see over here, separating from these cells, the whole area you call this a central cell, and central cell has two polar nuclei. So the polar nuclei plus the second male gamete forms the primary endosperm nucleus. And the primary endosperm nucleus basically again will divide later, will form the endosperm, which will be responsible for nourishing this particular zygote or basically the embryo. Okay. Now you have to explain like that. You have to give one by one. That is basically egg cell fuses with the first male gamete to form the zygote. Later the polar nuclei fuses with the second male gamete to form the primary endosperm nucleus. Now this process where the egg cell and the first male gamete is fusing, what do you call this? You call this as syngamy. Yeah, you call this as syngamy or you call this as fertilization. Okay, and now when over here the two polar nuclei and the second male gamete is fusing. So two nuclei is there, one nuclei is coming from the male gamete. So how many nuclei in total? 3 nuclei. So since 3 nuclei are coming together, you call this as triple fusion. And now since fertilization has taken place twice, over here also fusion has taken place, over here also fusion has taken place, you call this whole process as double fertilization. You call this whole process as double fertilization, children. Okay. So in 2 marks you can explain like that. So one is syngamy where this happens. Second is triple fusion where all this thing happens. Together what do you call them? Double fertilization. End product is this. Okay. Now what is a ploidy of antipodal cell and the primary endosperm nucleus? Ploidy as in the number of chromosome. Okay. So what do you have? You know all of them basically over here are haploid in nature. Because this is a result of meiosis. Megaspore mother cell underwent meiosis and then mitosis. So meiosis is underwent, right? So all of them were N. Now when it underwent mitosis, again it will be NN only. Okay. So ploidy of antipodal cell is N. But what about primary endosperm nucleus? So primary endosperm nucleus has, okay. So in the case of primary endosperm nucleus, I am writing it over here. Primary endosperm nucleus. So it has 2 polar nuclei. 2 polar nuclei were there plus the male gamete. 2 polar nuclei plus the male gamete. So how many nuclei? So this nuclei, this nuclei, this nuclei. So 1, 2, 3. Everyone has an N. How much N comes together to form 3N. So what is the ploidy, children? Triploid. It is triploid. Okay. This is haploid. Rest all of them are haploid. Just the primary endosperm nucleus becomes triploid. Okay. If you are asked what is a ploidy of 1 polar nuclei, it is just N. It is just N. Okay. Understand. What are the ploidy of 2 polar nuclei together? It is N and separate. Okay. Now, why the endosperm development precedes embryo development? Why do you think endosperm? Okay, now one more thing. So over here I said primary endosperm nucleus forms. This primary endosperm nucleus will undergo thorough division and then later form the endosperm. This thing will form the endosperm layer. Why do you think that endosperm forms first and then the zygote, which is already formed, it will form the embryo? Why is it so? Because the endosperm is the one that will provide nourishment for the zygote to actually form the embryo. That is why the endosperm forms first. Okay, so you got your 3 answers and you got your 5 marks as well. Now going ahead, 28. Picture related to pollination is given below. Okay, so there's an insect and the pollination is happening. Will this pollination confirm fertilization? Can you tell me the answer? Do you think that this type of pollination will confirm fertilization? No. What do you understand by pollination? You have to talk about that first. So pollination is when transfer of pollen happens from the anther to the stigma. Agree? To the stigma. This is pollination. Fertilization is what? You have to explain that now. Fertilization is when the male and the female gamete fuses. When the male and the gamete fuses. So pollination has happened, it's fine. But the pollen grain now has to form the pollen tube. Okay, so for that pollen pistil interaction should be right. Pollen pistil interaction should be there. It will later should be forming pollen tube. After forming pollen tube, the male gamete should be fusing. That's when I can say fertilization can take place. So how do you frame this answer? You will explain this, and you'll say this is the condition for fertilization to happen. Now, I cannot expect that every time a pollen grain lands on it, it will lead to the formation of pollen tube, because obviously the plants will be showing outbreeding devices, like it might show self-incompatibility or things like that. So I cannot confirm pollination. So I cannot confirm fertilization happening over there. So you have to explain that in points. Okay. Don't write too much. Don't write too much. Very limited. Be very limited. Be very specific. Underline what do you think is important. Underline pollen pistil interaction. Underline formation of pollen tube. Do not underline the whole thing there. Okay. Now, what are the floral rewards provided by the plants to the insects to revisit? What do they do? They give nectar. They give nectar. Sometimes they give shelter also. Sometimes they give shelter and everything. So you can write like that. Okay. Next, mention the characteristics of a flower in this pollination other than the floral rewards. So you know this is an insect, this is a flower. So obviously insect pollinated flowers have their own features. What are those? They should be looking they should be fragrant. Okay, they should be fragrant. They should be having large flowers so that it will be visible. They should be colorful, brightly colored. Okay, pollen should be sticky. Pollen should be what? Sticky, so that it sticks onto the surface of the insect that is pollinating. All these things. You can write it, right? Easy things are there. Fine? Works? Okay. Yes. Next question. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. Comment. So this is the same thing, the outbreeding devices. I already told you, but I'll explain it to you again. You have few more. Okay, what all did we already covered? In some species, pollen release and the stigma receptivity are not synchronized, as in the maturation is not happening properly. Okay, maturation at different times. Maturation at different times. This is known as dichogamy. I told you when male thing comes first, it is known as protoandry, or if the female matures first, it is known as protogyny. Okay. Together you call it dichogamy. If you want to learn it, you can learn it. It's for neat. If you do not want, just understand this term. If both of them are maturing at different times, obviously you cannot expect the fertilization or the pollination kit to take place properly. Okay. Now the anther and the stigma are placed at different positions that can be also taken place, such that it cannot come together. So for example, just think that over here the stigma is at this particular position, but the anther is chuttusa. Do you think that this can fly over and come over here? Very difficult. So that is the next step. The anther and the stigma are placed at different position. Now the third thing is self-incompatibility. Okay, self-incompatibility, I already told you, this is genetic mechanism. You call it self-sterility or interspecific incompatibility as well, in which the pollen pistil interaction does not take place, which is why the pollen tube formation would not be taking place. So over here they have written exactly like this. This is genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting the pollen germination or pollen tube growth. So pollens cannot germinate. Without germinating, can they form the tube? No. So pollen germination and pollen tube formation, everything is stopped. So you underline both of them, both the points, and then explain your answer. Another device is to produce unisexual flowers. Unisexual flowers as in a flower which just has one sex. So over here I will be just having the female one, not the male one. So that, or probably another flower which just has the male one, not the female one. This is what you mean by unisexual flowers. So you have 4 points. You have to explain them in whatever ways I have explained you. You can give the example of diagrams and explain if you want. Okay, and also mention it's a genetic thing and everything else works. So that's how you can okay. So in this way they will be discouraging self-pollination and explaining cross-pollination. You can explain what is cross-pollination and self-pollination as well in the end, okay, or in the beginning only. You can say self-pollination is when from the pollen of the same plant, the stigma from the pollen of the same flower to the stigma of the same flower, it is landing. This is known as self-pollination. Okay, cross-pollination is when from the from one flower to another flower of the same plant, or from one flower of one plant to another flower of another plant. If pollination is happening, this is known as cross-pollination. Okay, so self-pollination is inhibited because this could lead to inbreeding depression. Okay, write that point. It could lead to inbreeding depression. Hence, there are certain outbreeding devices, and those are as follows, and then answer your question. First, now the last question that we have over here is, mention the site where fertilization of the ovum occurs in the case of a female, human female. Tell me children, what is the site where fertilization occurs? So basically in the fallopian tube that you see over here, this is where the fallopian tube is the place where fertilization takes place. If you are asked about the exact place, so fallopian tube have 3 regions. One is having finger-like projection. This is known as fimbriae, but this area is known as infundibulum. Infundibulum. This area just above, just later to it, it is known as, where should I? Okay. So over here, this is fimbriae. This, okay. This area from here to here, you can call it as ampulla, and this area is known as isthmus. Isthmus is a narrow region, ampulla is a broad region. So fertilization happens in the ampulla. Always remember, it happens in the ampulla. So many years back it was ampullary isthmic region, it was between this region, but now NCERT has mentioned just ampulla, so remember that. Now explain the process of fertilization and mention how polyspermy is prevented. What do you mean by fertilization? Process of fertilization is basically when the female gamete the male gamete is being released into the female genital tract, and the male gamete later travels and fuses with the female gamete which is there in the fallopian tube. This is the fertilization. And how polyspermy is prevented, that is the question. So how is polyspermy prevented, my dear children? So polyspermy, what do you mean by polyspermy? Polyspermy basically means many sperms. We do not want the entry of many sperms. So how can you prevent the entry of many sperm? Only one female and the male gamete has to fuse, right? So first thing that happens is, understand this thing, okay? So you have this, agree? What is this? This is the female gamete. So I'm just, and it has a layer outside it. You call this layer as okay. You call this layer as zona pellucida. Okay. Now the zona pellucida layer has certain receptors over it. You call this as ZP3 receptors. You call this as ZP3 receptors. Now ZP3 receptors is where this sperm will actually come and bind. Okay, the sperm, they come and bind on this ZP3 receptors, and then what happens is that the sperm's head will be having, as I said, acrosome is there. The acrosome opens up. The sperm head, if you see, it looks like this. So this is the nucleus and everything. At the top you have the acrosome with all the enzymes. You call them lysins. So they are going to lyse open this zona pellucida and get inside the zona pellucida. So over here you will be having the sperm. Just imagine I am making a sperm over here. It does not look like a sperm, but yeah. Okay. One came in, and now it does not want another sperms to come inside. So what is the thing that happens here? So basically, when this sperm comes and attaches on the membrane of the secondary oocyte, what happens is that there will be certain channels that opens up. Certain ion channels that opens up. So what has taken place, children? First thing, the sperm sperm came and attached to the ZP3 receptors, and then the second thing that happened is acrosome reaction, and the sperms are then interacting. Sperm interact with membrane proteins. I'm not writing the name of the proteins. Okay, it interacts with membrane proteins, and now what has happened? The membrane channels, ion channels open up, and now what will happen? The Na, sodium ions comes inside. The sodium ions comes inside in a way that the membrane is now depolarized. The membrane will get depolarized. It will bring changes in the membrane such that any sperm that will try to come and enter here, they won't be getting entry. So this is known as fast block polyspermy. This process that you just saw right now, where the sperm came, attached, the proteins interacted, and now the ion channels opened up, the Na got inside, the sodium ions got inside, and it causes depolarization of the membrane, which is basically change in the membrane such that the sperms cannot enter. You call this as fast block polyspermy. Fine. This is one thing, how you can block. Now there is something that is known as slow block polyspermy. Just after this, next thing gets activated. This is temporary, okay? This is temporary, obviously. So next thing get activated, that is basically, now when the nucleus gets inside, here you have the cell, normal cell is there, secondary oocyte is a cell, right? So it will be having its own organelles, and one such organelle is, children, the smooth endoplasmic reticulum. One such organelle is smooth endoplasmic reticulum, which has lots and lots of calcium, which has what? Calcium ions. Now what will happen? Because of all this happening, calcium ions moves outside. Calcium ions, they moves outside, and when calcium ions moves outside, there comes the role of next thing that is over here. So there are certain granules also in the cytoplasm, and these granules have certain enzymes. So this Ca, that is calcium ions, they slowly go and tell the granules, hey granules, go fuse with the membrane. So these granules will go fuse with the membrane and release all the enzymes over here. This enzyme that you see that will be released over here from the granules, they will make all the ZP3 receptors to be cut. So everywhere, wherever there are ZP3 receptors, they will be cut. So what will happen? Basically, no other sperm can identify and they will try to come inside. This is known as slow block polyspermy. What has happened, children? The smooth endoplasmic reticulum, it had calcium ions. The calcium ions basically told the cortical granules, these granules that you see over here, they are known as cortical granules, to go and fuse with the membrane and release all the enzymes such that the ZP3 receptors that are over here can be taken down and no other sperms can get inside. Now this is known as slow block polyspermy. Okay, now I have not written the answer in such format. Okay, I have the answer for the second part of the question. If you want me to write the answer and give it to you, you can tell me in the comment section that please ma'am, as you said, can you provide the polyspermy answer as well? So I will write it and give it to you. That's my wada, promise. Okay. Got it? But I want you to try it for sure. So this is very, 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 very easy. It's not very difficult, humongous thing. You have to know how to write the answers. Yeah. Now name the embryonic stage that gets implanted in the uterus. Now which stage? Embryo has different stages, I told you. Okay, so 2 cell, 4 cell, 8 cell, and then goes on. So it is at which stage that the implantation takes place, children? It is known as blastocyst stage. Blastocyst stage. In blastocyst stage, what happens? Basically, it has 100 to 120 blastomeres, and over here there will be an outer layer like this. Okay, you call this layer as trophoblast. You call this layer as trophoblast, and there will be a layer like this inside, which is known as inner cell mass. So it is at this stage that the implantation takes place in the uterus, and here is the answer for the same. So you can explain the process of implantation in human females. So basically, after fertilization, the zygote undergoes several division. What happens? There is a structure that is being formed, which is known as blastocyst, which is about 5 to 6 days after fertilization. The blastocyst consists of an outer layer that is known as trophoblast, which forms the form of placenta. So outer layer, that is trophoblast, help in the formation of placenta, whereas the inner cell mass will be developing into the embryo later on. Even though it's the embryo itself, but still we say it will be the one that will be growing to a fetus or embryo later on. Now, movement to the uterus. The blastocyst travels down from the fallopian tube towards the uterus. It's not like that, so I'm just changing it. Okay. Now over here, attachment. I will write around 6 to 7 days after fertilization, the blastocyst reaches the uterus and begins the process of implantation by attaching to the endometrium. So yes, blastocyst, it is being formed. So morula is being divided. Morula is when it has 8 to 16 cells. Okay, at this point of time, it slowly divides, and that's when it forms blastocyst. So it is slowly traveling towards the uterus itself, but it's towards the uterus. Okay, and now it attaches on these days. Okay, and now what will happen? Begins the process of implantation by attaching to the endometrium, that is the uterine lining. Fine. Now what will happen? The trophoblast cells starts to proliferate. The trophoblast that you see over there, that is the outer layer, it will slowly proliferate, it will divide itself and invade the uterine lining. It will get inside the uterine lining. So uterus is over here. Uterus is over here, children. Okay. The trophoblast has come. It slowly tries to invade into the uterine lining. Okay, the trophoblast starts to proliferate and invade the uterine lining, secreting enzymes that breaks down the endometrial tissues, allowing the blastocyst to embed itself into the uterine wall. Okay, the implanted blastocyst starts forming placenta, which will provide nutrients and oxygen to the developing embryo. So there is also one more thing to given over here in your book, that is chorionic villi. Chorionic villi. So what happens is that when this implantation and everything takes place, another layer, so like this is formed. Okay, like this, like hands, it is being formed, which goes and attaches. So this type of villi that you see is known as chorionic villi. So this is formed with the help of the trophoblast. Okay, this is formed with the help of the trophoblast and also the maternal tissue. So what do you say? How do you say the formation has taken place? Basically, the trophoblast undergoes formation, and also the maternal uterine wall also have changes such that both of them they embed and form the whole thing, that is the placenta. That's how you have to explain your answer. So 1, 2, 3, 4 points in points, try to explain. See how many marks this is contained. If it's of 3 marks, don't go explaining a lot. Give priority to the other part. But if it's of more marks, then explain about chorionic villi. Okay, how the trophoblast is now going to form the chorionic villi. What is the role of the maternal tissue also in the formation of placenta. So always remember, placenta is formed not just with the help of the fetus. It's basically the growing embryo as well as the maternal uterine tissue which comes together to form the placenta. Okay, done. So that's all, children. As you can see, I am also kind of tired, and I know that pretty sure that you also might be tired, but it would be worth it. It contains all the important questions, and in and around I was trying to explain you a lot of things, okay, which might come for your exam. So I just wish you all the best. Go through your NCERT, and once you're done with the NCERT, at least one portion, just let me know in the comment section that, ma'am, we are done with human reproduction, we are done with reproductive health. I'll suggest start with human reproduction. then go for reproductive health, complete it. Okay, it's easy. Or else if you, whenever you do reproductive health, before that you have to do human reproduction. That is the first thing. So either you start with sexual reproduction in flowering plants, that is a completely different topic. So you can start with that and then go for human reproduction and then reproductive health. Go like that. Okay. And once you're done with the NCERT, do let me know. So that's all, children. I hope that this was helpful and I wish you all the best. Take care and all the best for your midterm examination. Be happy.